Hello, this is Mr. White. We are on Chapter 8. This is Lesson 3, Advances in Genetics. My Planet Diary. Zorses and Zedons. Most people can tell the difference between a zebra and a horse, but would you be able to tell the difference between a zorse and a zedonk? Both types of animals are zebroids or zebra hybrids. These animals result when a zebra mates with a horse or a donkey. Zebroids do not usually occur in nature. They generally result when people cross them on purpose. People may have first crossed zebras and horses in an effort to develop disease-resistant transportation animals for use in Africa. Zebras are resistant to African sleeping sickness. It was hoped that zorses, the offspring of zebra and horses, would have the resistance. How can organisms be produced with a desired trait? Unless you're an identical twin, your DNA is different from everyone else's. Because of advances in genetics, DNA evidence can show many things, such as family relationships or the ability to produce organisms with desirable traits. Selective breeding, cloning, and genetic engineering are three different methods for developing organisms with desired traits. Selective breeding. The process of selecting organisms with desired traits to be parents of the next generation is called selective breeding. Thousands of years ago, in what is now Mexico, the food that we call corn was developed this way. Every year, farmers saved seeds from the healthiest plants that produced the best food. In the spring, they planted only those seeds. This process was repeated over and over. In time, farmers developed plants that produced better corn. People have used selective breeding with many types of plants and animals. Two techniques for selective breeding are inbreeding and hybridization. Inbreeding. The technique of inbreeding involves crossing two individuals that have similar desirable characteristics. Suppose a male and a female golden retriever are both friendly and have the same coloring. Their offspring will probably also have those qualities. Inbreeding produces organisms that are genetically very similar. When inbred organisms are mated, the chances of their offspring inheriting two recessive alleles increases. This can lead to genetic disorders. For example, inherited hip problems are common in golden retrievers and other types of inbred dogs. Hybridization. In hybridization, breeders cross two genetically different individuals. Recall that a hybrid organism has two different alleles for a trait. The hybrid organism that results is bred to have the best traits from both parents. For example, a farmer might cross corn that produces many kernels with corn that is resistant to disease. The farmer is hoping to produce a hybrid corn plant with both the desired traits. Roses and other types of flowers are also commonly crossed. Cloning. For some organisms, such as the dog shown in Figure 1, a technique called cloning can be used to produce offspring with desired traits. A clone is an organism that has exactly the same genes as the organism from which it was produced. It isn't hard to clone some kinds of plants, such as African violets. Just cut a stem from one plant and put the stem in soil. Water it, and soon you will have a whole new plant. The new plant is genetically identical to the plant from which the stem was cut. Genetic engineering. Geneticists have developed another powerful technique for producing organisms with desired traits. In this process, called genetic engineering, genes from one organism are transferred into the DNA of another organism. Genetic engineering can produce medicines and improve food crops. Genetic engineering in bacteria. One type of bacterium is genetically engineered to produce a human protein called insulin. Many people with diabetes need insulin injections. Bacteria have a single DNA molecule in the cytoplasm. Some bacterial cells also contain small circular pieces of DNA called plasmids. You can see how scientists insert the DNA for the human insulin gene into the plasmid of a bacterium in Figure 2. Once the gene is inserted into the plasmid, the bacterial cell and all of its offspring will contain this human gene. As a result, the bacteria produce the protein that the human gene codes for, in this case insulin. Because bacteria can reproduce quickly, large amounts of insulin can be produced in a short time. Small rings of DNA, or plasmids, can be found in some bacterial cells. Scientists remove the plasmid. An enzyme cuts open the plasmid and removes the human insulin gene from its chromosome. The human insulin gene attaches to the open ends of the plasmid to form a closed ring. Some bacterial cells take up the plasmids that have the insulin gene. When the cell reproduces, the new cells will contain copies of the engineered plasmid. The foreign gene directs the cells to produce human insulin. Genetic engineering in other organisms. Scientists can also use genetic engineering techniques to insert genes into animals. For example, human genes can be inserted into the cells of cows. The cows then produce milk containing the human protein coded by the gene. Scientists have used this technique to produce the blood-clotting protein needed by people with hemophilia. Genes have also been inserted into the cells of plants, such as tomatoes and rice. Some of these genes enable the plants to survive in cold temperatures or in poor soil. Other genetically engineered crops can resist insect pests or contain more nutrients. Gene therapy. Someday, it may be possible to use genetic engineering to correct some genetic disorders in humans. This process, called gene therapy, will involve inserting copies of a gene directly into a person's cells. For example, doctors may be able to treat hemophilia by replacing the defective allele on the X chromosome. The inserted gene would provide the body the correct instructions to clot blood normally. Concerns about genetic engineering. Some people are concerned about the long-term effects of genetic engineering. For example, some people think that genetically engineered crops may not be entirely safe. People fear that these crops may harm the environment or cause health problems in humans. To address such concerns, scientists are studying the effects of genetic engineering.